Many of us have learned about DNA and genes even through, a New York Times article or something like that. And so I think we all have an understanding that there's this recipe of letters that somehow generates the protein or the building blocks of our body. And that's in every cell and every part of our body. But what's interesting about that concept of genes and DNA is my kidney cells have the same recipe as my eyeball cells. Yet they do very, very different things. So how do we take that same recipe and come up with very different purposes? And so epigenetics is one of the body's mechanisms for doing that. And the term epigenetics literally means on top of genes. So what has to happen for that recipe to give you something very different to make an eyeball than to make a kidney is you have to pretend it's like a recipe book. You have to know which page to go to and turn on the recipe for apple pie versus the recipe for turkey. And so those mechanisms that say, go to this page and turn that gene on, if you will, are epigenetic mechanisms. And there are many different kinds of sort of chemistry involved, but the general concept is, how do you know where in the genome to go and turn something on to do the purpose of that cell that might be different than the purpose for some other kind of cell? You imagine a recipe book where you had tabs for those pages. So when you were looking up apple pie, maybe there's a blue tab, and when you're looking up turkey, there's a green tab, and it's in a specific location on the book. Environmental factors can mess with those. Just like if you throw the recipe book around it, you could wear thin that particular tab, and it wouldn't be as useful to quickly get you where you want to be. And so it's a simplification, but I think that analogy really works, that these tags, if you will, can be affected by environment. And when they are, that can really muck up how easy it is to do this kind of quick finding and regulation of turning particular genes on and off. If we understand that there are particular chemicals that mess with particular tags, that could be quite useful. First, it would tell us where to go in that recipe book or where to go in the genome to understand maybe something about the mechanism of that particular toxicant or chemical. The other thing it can do is tell us where to go to look for changes. If something has gone wrong and we want to show that there's been a biological effect of a particular toxicant, knowing where to go and look could be quite useful. Right now, what we do is we look for the what we would say phenotype or presentation, whatever the biological entity might be, which is important. But if we wanted molecular representation of response, this might be one opportunity to do that easily. In environmental health, I think that the sort of work around epigenetics has at least two purposes. And so one is this idea that we might understand mechanism better. So if we want to understand why a particular toxicant or chemical creates a biological problem, if it's through an epigenetic mechanism, we've learned a lot more about the biology that we may then be able to intervene on. The other is as a biomarker. Even if the epigenetic marks are not part of the story of why a chemical causes a health outcome, even if that's not true, if those marks are a good proxy for the chemical exposure, then we would have a really good biomarker of what has happened in the past. And that can be useful if you're trying to understand dosing and those kinds of things, or in the sort of cumulative environmental exposure, if you're trying to get a biomarker that isn't specific to a particular chemical but is kind of a gross metric of those. That biomarker world could really move forward what we're able to do as epidemiologists and likely as toxicologists as well. How does this inform toxicology, or the worlds that I live in often, or how does this inform environmental health or epidemiology? And unless you bring folks together from those different worlds who understand what they could use to translate these kinds of findings, to talk to the people who are developing the epigenetic measurement techniques and to talk to the people that are developing the biological story and understanding, everybody's going to continue to work in silos, and they may never meet up and sort of answer each other's questions. And so meetings like this where people can come together, I think really helps you at the beginning of a nascent field to get it together in the right direction.